Warm greetings to all. I am D. Karthika, Ph.D. scholar working under the guidance of Dr. G. Velmurugan, scientist, KMCH Research Foundation, and also under the co-guidance of Dr. Shankar Jha, assistant professor from School of Chemical and Biotechnology at Shasta University. My PhD title is on profiling of gut microbiota and its metabolites during endocrine-disturbing chemical-induced glucose dyshomeostasis, implication of host-glucose metabolism. Over the past few decades, there has been a global transition in the disease pattern, but non-communicable diseases, particularly type 2 diabetes, became a major health burden. At the same time, the environmental exposure that has significantly increased due to the industrialization and the modern lifestyle. Although that the chemicals are known to interfere with the hormonal balance, the mechanisms by which they contribute to the metabolic disorders such as diabetes are not fully understood. There are emerging evidences suggest that the gut microbiota may play a major role in the mediating of these effects. The problem statement is chronic low-dose exposure to the environmental disturbing chemical may contribute to the diabetes via the gut microbiota alterations. In this study, we adapted an integrated approach of meta-analysis in vivo mouse models and fecal microbiota transplantation experiments. We also simulated real-world exposure by administering the mixture of multiple endocrine-disturbing chemicals at environmentally relevant doses. To better understand the metabolic effects that the chemical exposed mice were compared with the high-fat diet models, but both groups developed the glucose intolerance, but the high-fat group showed the obesity. In contrast, that the chemical exposed resulted in a distinct non-obese diabetic phenotypic pattern. In analysis of gut microbiota revealed a consistent and the reproducible pattern of dysbiosis in the chemical exposed animals. This includes a reduction in the firmicutes to the bacterials ratio and a depletion of beneficial short-chain fatty acid producing bacteria. At the same time, there was a significant increase in proteobacteria, bacteria, and notably desulfobacteriota are microbial groups commonly associated with the inflammations and the environmental stress. One of the most striking findings was the enrichment of desulfobacteriota, which are sulfate-reducing bacteria known to produce hydrogen sulfide. The excess of hydrogen sulfide can impair the mitochondrial functions and that leads to the disruption of intestinal barrier and promote inflammations and also alter the host glucose metabolism, thereby contributing the metabolic dysfunction. Interestingly, this microbial signature was distinct from the observed in the high-fat diet groups where the desulfobacteriota levels were reduced. This suggests that the chemical-induced diabetes may rise through fundamentally different microbial mechanisms. This study suggests that the chemical-induced diabetes may rise through fundamentally different microbial mechanisms. These findings highlight that the potential of gut microbiota signatures, particularly desulfobacteriota and the microbial hydrogen sulfide, which is known as a novel biomarkers for disease detection and risk stratification. In conclusion, the study demonstrates that the chemical exposure can reshape the gut microbiota and drive the non-obese diabetes through microbiota-mediated mechanisms that could opening a new avenue for the precision medicine. The targeting these pathways through the microbiota modulations by inhibiting of sulfate-reducing bacteria or restoration of beneficial metabolites may offer new therapeutic strategies for the diabetes management. The future therapeutic strategies in the gut microbiome research focusing on restoring of beneficial bacteria and corresponding the dysbiosis to combat the metabolic diseases. And the key approaches include that the targeting of the sulfovibrio-related H2S biomarkers that inhibiting the sulfate-reducing bacteria and restoring the short-chain fatty acids via the microbiota targeted therapeutics. I need to acknowledge for the funding agencies for the financial support for pursuing my research, and I also deeply extend my thanks to all the team members of KMCH Research Foundations and supporters. Thank you all.